welcome back to Learning with Isha. In this series, we're talking about respiration. We have already looked at what is respiration, where it takes place, and what is the amount of energy released, and how it is stored. In this video, we're going to talk about the first step of cellular respiration. This step is known as glycolysis. It is common both in the case of aerobic respiration and also in the case of anaerobic respiration. As we have already discussed, aerobic respiration is in the presence of oxygen and anaerobic is in the absence of oxygen. The further steps in both the cases are going to be different, but glycolysis is the common step in both these respiration pathways. So this is going to be the first step. In fact, in anaerobic respiration, this is going to be the only step that is going to release energy. We'll look at more about that in the upcoming videos. So let's begin talking about glycolysis. The word glycolysis is made up of two words, glycose, which means sugar, and lysis, as we already know, is known as breakdown. So glycolysis literally means the breakdown of sugar. So where is this breakdown of sugar taking place in the cell? Well, the location of glycolysis in the cell is the cytoplasm. So it takes place in the cytoplasm of the cell. Who discovered this pathway? Well, this pathway was illustrated by three scientists. The name of these scientists was Gustav Embden, Otto Meyerhoff, and J. Parnas. In the honor of these scientists and taking the initials of their name, as in E, M, and P, the glycolysis pathway is also known as the EMP pathway. So, in the glycolysis pathway, till now we've already come to understand that there is going to be some sugar that is going to be broken down. The sugar that is normally broken down in the case of glycolysis is glucose. This glucose, where does it come from? The sources are different both in the case of animals and plants. We know that in animals, the reserve food is stored in the form of a molecule which is known as glycogen. So this glycogen is going to be broken down, and from there we are going to get glucose. This is in the case of animals. What about plants? Where do plants get these glucose from? Well, plants have a molecule known as sucrose that serves as their storage material. Now, this sucrose molecule is a disaccharide. Disaccharide means it has two monomers. It is made up of two sugars. The two monomers that make up sucrose are glucose and fructose. So, glucose and fructose are the two molecules I am going to get by splitting sucrose. An enzyme that is going to cause this reaction, to cause this splitting, is known as invertase. So this invertase would break down the sucrose. The glucose would go into the glycolysis pathway. The fructose would also go into the pathway, and then the pathway is going to proceed. How many reactions in total will be present in the pathway? As we're going to look ahead, that a total of 10 reactions are going to be present in this pathway. While studying this pathway, what do we need to remember? We need to remember that in each step, what was the substrate and what would be the product. So number one is that what is the substrate and what is the product. Also take care of the fact which enzyme would be involved in each step, and furthermore also take care that whether any energy is being consumed in that step or any energy is being liberated in that step. So you also have to take care whether some ATP or NADH2 is being consumed or released. Now let's go ahead and actually start looking at the steps of this pathway. So the first step of glycolysis definitely has to begin with a glucose molecule. So there will be a glucose molecule that will get converted to another molecule. So glucose is the substrate. What is the product for the first reaction? The product is a molecule known as glucose 6-phosphate. So there is a phosphorylation at the 6th position. This is a phosphorylation reaction. So a phosphate group is being added. Where do we usually get phosphate groups from? We need to break down a molecule of ATP, and this will give me ADP and one inorganic phosphate. This phosphate group is going to get added here. So take care, one ATP is being used in this step. Now let's talk about the enzyme over here. The enzyme is an enzyme known as hexokinase. So this hexokinase is going to add a phosphate group. A cofactor for this enzyme is the magnesium ions. So it requires the presence of these ions to work optimally. Now the second step is the conversion of this glucose 6-phosphate into its isomer, that is fructose 6-phosphate. So this is an isomerization reaction, just a structural change, no energy required here. The enzyme involved in this case is going to be phosphoglucoisomerase. Phosphoglucoisomerase, as it is an isomerization reaction. Now, the next step, as in the third reaction, would be the conversion of this fructose 6-phosphate to another molecule known as fructose 1,6-biphosphate. So now I get fructose 1,6-biphosphate. We again see that a phosphate group, initially I had one phosphate group. I note that this is again a phosphorylation reaction. One more phosphate group is going to get added. For the addition of phosphate group, again I am going to require the conversion of ATP to ADP. This means that one ATP is consumed over here also. Now the enzyme in this case is going to be phosphofructokinase. So the kinase enzyme basically does what? It moves around a phosphate group. So here we have seen the initial steps, glucose getting converted to glucose 6-phosphate, a phosphorylation reaction. After that, glucose 6-phosphate getting converted to its isomer, fructose 6-phosphate. Another phosphorylation reaction where another phosphate group is added to this fructose. We saw that the sucrose was breaking down into glucose and fructose. The glucose can directly enter this pathway and the fructose can get phosphorylated and it can also enter this pathway. What is the aim of such phosphorylation reactions? These phosphorylation reactions activate the sugar. Why do we need this activation? So that the sugar does not escape the cell. Right? If I get one molecule of glucose, it might get used in the cell for another reaction and might not give energy. So if I require energy and I require these molecules to remain inside the cell and not escape, I need to phosphorylate them. Now let's move on to the next reaction. In the next step, this fructose 1,6-biphosphate or bisphosphate is basically, you see, a 6-carbon compound. We started with glucose was 6 carbon and furthermore there's just isomerization, okay? So the 6 carbon compounds are continuing. Here we will have the first splitting step. The 6-carbon compound fructose 1,6-biphosphate is going to split into two 3-carbon compounds. That is, first will be your 3-phosphoglyceraldehyde. The next molecule is going to be dihydroxyacetone phosphate. Both of them being 3-carbon compounds. We saw that fructose 1,6-biphosphate had two phosphate groups. One phosphate group is going to be divided in each molecule. One phosphate in dihydroxyacetone phosphate and one phosphate in 3-phosphoglyceraldehyde. The abbreviations used here are PGAL for this molecule and DHAP for this molecule. The enzyme that is responsible for the splitting reaction is known as aldolase. Now, something interesting happens here. Both these molecules are in fact isomers of each other. Only one of the molecule is going to further proceed in the reaction and the other one is going to get isomerized. So we see that only 3-phosphoglyceraldehyde would continue further in the reaction and dihydroxyacetone phosphate is going to get isomerized into this. So in the further steps when we continue, we would consider only one molecule of PGAL and everything whatever we obtain will be multiplied by 2. Why? Because there was supposed to be another reaction that took place with this 3-carbon compound. So now let's look at the next step. Now let's continue with this PGAL molecule and let's see what happens. Now this PGAL molecule is a 3-carbon compound. So we are looking at only one half of the reaction. So now this PGAL molecule is going to add an inorganic phosphate and get converted into 1,3-bisphospho or biphosphoglyceraldehyde. So here you see that we had only 3-phosphoglyceraldehyde, one phosphate group. In this case, another phosphate group is going to get added and this is going to get a bisphosphoglyceraldehyde molecule. In the next step, this aldehyde is going to undergo an oxidation reaction. So in this oxidation reaction, this is going to get converted into 1,3-bisphosphoglyceric acid. So the aldehyde just got oxidized to the glyceric acid form. The enzyme involved here is phosphotriose dehydrogenase because this is a dehydrogenation reaction, addition of oxygen and removal of hydrogen. Now, since we know in biological systems we have redox reactions. If there is oxidation of something, it must be coupled to the reduction of something. What is going to get reduced here? Well, we see that NAD plus gets reduced to NADH plus H plus. So we see the formation of NADH plus H plus in this step. In the next step, 1,3-bisphosphoglyceric acid is going to get converted into 3-phosphoglyceric acid. 3-phosphoglyceric acid, which is commonly known as 3-phosphoglycerate. Even here I could have written 1,3-bisphosphoglyceric acid or 1,3-bisphosphoglycerate. So here we write 3-phosphoglycerate. In the next step, this is going to get converted to 2-phosphoglycerate. You see that in this step, there is only a rearrangement of the phosphate group. An enzyme involved in this case is mutase. Mutase enzyme causes such rearrangements. Now this 2-phosphoglycerate is going to get converted into another molecule known as 2 phosphoenol pyruvate. Now, this phosphoenol pyruvate enol group is added by an enzyme known as enolase. In the last step, this 2-phosphoenol pyruvate will be converted to the final product of my glycolysis, that is pyruvic acid or pyruvate molecule. This is the final product of glycolysis, and the enzyme involved here is pyruvate kinase. Pyruvate kinase. Now you see that in this sequence of reactions, what is exactly happening? A phosphate group is being lost here. From 1,3-bis phosphor, we are coming to only one phosphor group at the third position. Where is this lost phosphate group going? This will be accepted by an ADP and form ATP. So for the first time in glycolysis, we see that ATP is being formed. Now in the next step, there's just a rearrangement. Here we see that again 2-phosphoenol pyruvate is being formed, and in the last step again a phosphate group is being lost. This molecule is losing the phosphate group. Again the same reaction is going to happen. ATP molecule is again going to be formed here. So we see that in the first half of the reaction, 2 ATP molecules were consumed. In the second half of the reaction, 2 ATP molecules are given out. But please take care that the second half of reaction is only for this molecule. There would have been a DHAP molecule that would have also gotten converted into PGAL. So whatever products you get here should be multiplied by 2. Why? Because I have to consider the other molecule as well. So now let's just go ahead and look at what do we finally get in glycolysis. Now let's just try to summarize this. We have seen that in glycolysis there are 2 steps in which the energy is consumed. In the first half of the reaction, we were consuming 2 ATP molecules for the phosphorylation steps. So this phase, the initial half, is the energy consuming phase of the glycolysis. Why? Because energy is being consumed here. ATP is being used to perform the phosphorylation reactions. Whereas in the second half of the glycolysis reaction, there are 2 energy yielding steps. Now, this is known as the payoff phase. Why? Because this glucose breakdown is paying me some kind of energy currency. So this is the second phase, that is the payoff phase. In the payoff phase, we see that there are 2 steps at which 2 molecules of ATP are liberated. One, when you see that 1,3-biphosphoglyceraldehyde is getting changed to 1,3-biphosphoglycerate. In this step, you see what is happening, an aldehyde is getting converted to an acid. Glycerate means phosphoglyceric acid. So this represents aldehyde to acid is a oxidation reaction. But we know that in biological systems, we don't have oxidation and reduction. Rather, we have redox reactions. So for every oxidation, there has to be a reduction. What is the reduction? You see, the NAD plus is getting reduced to form NADH plus H plus. So what we get is one molecule of NADH plus H plus. Besides that, in the next step, when 1,3-biphosphoglycerate is getting converted to 3-phosphoglycerate, you see there are 2 phosphate groups here at the first position and at the third position. One of the phosphate groups is going to get removed. This phosphate group is going to convert ADP to ATP. So one ATP is produced here. Similarly, in the step where phosphoenol pyruvate is converted to pyruvate, so one phosphate group of the phosphoenol part is getting liberated. Again, that is going to give me ATP. But keep in mind that this is only from the PGAL molecule. We know that the DHAP is also going to get isomerized into PgAl. So the same reaction is going to give me 1 ATP here also and 1 ATP here also. So in the payoff phase, therefore, we are going to gain 4 ATP and 2 NADH plus H plus. Let's try to summarize this. So the energy ATP consumed was 2 molecules as we saw, and the ATP formed were 4 molecules. And we also saw that 2 molecules of NADH plus H plus were formed. So eventually we see that there is a net gain of only 2 ATP molecules. Minus 2 here, plus 4 here. So therefore only 2 ATP molecules are gained. If you had to write a reaction for the glycolysis process, how would it look like? It would look something like this. See, what are you giving in the reaction? You're giving a molecule of glucose. It's a 6 carbon compound, and this is getting converted to 2 molecules of pyruvate in the end. The pyruvate is actually a 3 carbon compound, so 3 into 2, 6. Yes, this is right. Now we see that 2 ATP molecules are formed. To form these 2 ATP, we need 2 ADP and 2 inorganic phosphates, right? And we also see that 2 NADH plus H plus are formed. So yeah, let's just state that 2 NADH plus H plus. For this, we required 2 NAD plus molecules. And during the glycolysis process, eventually, we get this result. Well, this brings us to the conclusion of this video on glycolysis. In this video, we not only talked about the steps of the glycolysis, but also understood what are the energy carriers that are formed as a result of this process. Now, in the next steps, it would be very suitable to look at these pyruvate molecules are formed, but what exactly is going to be the fate of these pyruvate molecules? You see, glycolysis is not a complete oxidation. Complete oxidation कब बोलते हैं? अगर 6 carbon compound glucose would have been broken down into 6 carbon dioxide and released. So this is complete breakdown. Here we see that there is a partial breakdown. Why? A 6 carbon compound glucose is broken into 2 3 carbon molecules of pyruvate. So glycolysis actually is a partial oxidation. So in the next step, let's look at what happens with these pyruvate molecules. Thanks for watching the video. Don't forget to subscribe to my channel and like this video. See you.